Unlike the B cells that we saw in the humoral defense mechanisms, T cells do not release antibodies. T cells are better suited for a cell-to-cell kind of interaction. That is, they form the basis for our cellular defense mechanisms. Some T cells directly destroy antigenic cells and antigens, while others release chemicals that help regulate the immune response. There are two main classes of T cells. There are T cells that get activated from what's called a CD4 kind of precursor. The CD4 is an inactive cell, but when it becomes activated, it usually forms a helper T cell. Although some CD4 cells, when activated, become regulatory T cells to help moderate the immune response. There's also a CD8 kind of inactive T cell that when it becomes activated becomes the cytotoxic type of T cell, the kind of cell that acts directly in the destruction of pathogens and antigens that might be in the body. Unlike B cells and antibodies, T cells cannot see free antigens. T cells can only respond to an antigen fragment that is presented by antigen-presenting cells, or APCs. APCs present these fragments attached to MHC proteins that we discussed in a previous discussion. MHC proteins come in two different classes. The class 1 kind of MHC protein is found on the surface of all body cells except for blood cells. These class 1 MHC proteins present endogenous types of antigens, meaning antigens that are produced within the cell that's going to present them. That can be because of an intracellular kind of bacteria, or an intracellular kind of virus, or even certain cancerous kinds of cells. These Class 1 MHC proteins typically activate the CD8 type of precursor cell that becomes a cytotoxic T cell. Here, Class 1 MHC proteins are activating a cell-to-cell kind of defense mechanism. Class 2 MHC proteins, on the other hand, are found on dendritic cells, macrophages, and B cells, meaning they're found on the major kinds of APCs. They present exogenous antigens, meaning antigens that come from an outside source, like antigens that were flowing freely through the circulatory system, or antigens that are caused by a cell or a virus outside of the cell. These class 2 MHC proteins activate the CD4 kind of precursor cells, the ones that either become helper T cells or regulatory T cells. T cells can only be activated by antigen-preventing cells, and this activation is a two-step process. The first step is the antigen binding. Receptors on the T cell bind to an antigen MHC complex. Also keep in mind that each T cell has receptors that are all of the same three-dimensional shape. Each T cell will only be able to respond to one kind of antigen, specifically to one kind of antigen MHC complex. The second step in the process is called co-stimulation. In co-stimulation, chemicals on the surface of the APC that was presenting the MHC protein can cause the T cell to activate and proliferate and make more copies of itself. This cellular proliferation peaks at about 1 week after infection by the antigen. And then from 7 to 30 days after activation, the T cells undergo a process called apoptosis, meaning they destroy themselves. This step is necessary so that we're not under a constant kind of defense response, which would maybe cause constant fever and constant inflammation. We need those other symptoms of infection to subside after we've been able to successfully fight off that infection. While they are circulating, each T cell plays a specific role in the immune response. The helper type of T cell helps the immune response by activating B cells to produce antibodies, by activating the cytotoxic CD8 types of T cells, and by amplifying the innate immune responses, like activating macrophages, increasing inflammation, or even causing fever. The cytotoxic type of T cell, those that develop out of an activated CD8 precursor, directly attack and destroy other cells. The main targets are cells affected by intracellular pathogens like viruses, bacteria, and even certain kinds of cancers. The third class of T cells are called the regulatory T cells. They help dampen and moderate the immune response. This helps to suppress any self-reactive kinds of lymphocytes to prevent autoimmune reactions, to prevent our own T cells from recognizing and trying to attack our own body cells. Natural killer cells, macrophages, antibodies, and T cells all act vigorously against any kind of non-self cell, anything that is showing an antigen that does not belong to the body. If we're trying to do an organ transplant or a tissue transplant, it's necessary to find a donor with a similar ABO blood group, as well as similar MHC antigens. Keep in mind that there are millions of combinations of these antigens, so there are millions of possible combinations, and we need to try to find the best donor. Because the more different the combination of antigens are, the more likely the organ will get rejected by activating an immune response. The immune system doesn't know that a new kidney is there to help with the formation of urine and controlling body fluids. It only recognizes that the MHC antigens on the surface of those cells don't belong there, and it might be some kind of massive infection. There are 4 types of grafts or transplants that can be done, and in each one we have to try to find the best kind of match for these MHC antigens. The first kind of graft is called an autograft, a self-graft, because transplants are taken from the patient's own body. This is mostly done in the skin. If we're trying to remove a portion of a skin in one place to do something like graft over a burn on the body, we can graft that from the patient's own skin. Unfortunately, most of us don't have a backup heart or a backup brain or backup kidneys and livers, so we can't really do an autograft for most of those internal body organs. Because of that, the next best fit is what's called an isograft, transplants from a genetically identical person. And because they're genetically identical, they should also have identical MHC and ABO antigens. This is only possible in cases where somebody has an identical twin, which is relatively rare. So the most common kind of graft are called allografts. These are transplants from non-genetically identical people. Maybe it's another sibling, maybe it's a total stranger to the person, but we're trying to find the best fit for these MHC proteins. However, because there are millions of possible combinations of MHCs and ABOs, it's unlikely that we're going to find an exact match between a donor and a recipient. Because of that, in most cases where a person has received a graft or a transplant, there's likely going to be an immune response. And so patients are usually prescribed immunosuppressants, meaning that it helps prevent the rejection of the organ transplant, but it also makes them more susceptible to succumbing to another kind of infection. Folks who have allografts are not only in danger of still potentially rejecting the organ transplant, but are now also at increased risk for things like the flu. The fourth kind of graft is called a xenograft, or an outside graft. These are transplants from another species. Because most mammals are very similar on a genetic level, they do also have similar kinds of MHC and ABO antigens. But because they are less similar to humans, it's even less likely that we'll find a really good match in this other species. But it can be done in certain cases. The most common is a transplant involving the heart valves from a pig being transplanted into the heart of a human.